All right, let's start. So, so this is the SDS-PAGE lecture. I'm going to cover how the electrophoresis works, and also talk a little bit about glycine, because you probably have wondered how the glycine works in terms of shielding proteins so that it would resolve proteins better in the resolving gel. And also how the protein is separated, and also how to determine the molecular weight using molecular weight markers based on mobility, right? So, and also briefly, I'm going to briefly talk about how to read each lane. So let's start. So, um, So SDS-PAGE, basically, if you look at SDS-PAGE, if you look at right here, you have, sulfate. So, and then sodium sulfate of some sort. And then, there is the, dodecyl component right here. So, again, SDS, is the sodium sulfate of some sort. In this case, dodecyl, you have 12 carbons, right? So anyways, and then you have, PAGE, it's a polyacrylamide and gel electrophoresis. So again, that's what SDS-PAGE is. Oh, by the way, I just noticed I put out about the previous slide stuff on top. By the way, you didn't have to draw the structure. I mean, if you want to draw, that's fine. But the half of this, the screen is used for the previous slide for the most part. This way, you don't have to stop the video. So hopefully you could take notes as we go along, right? And when I talk about stuff, I'm probably diluting stuff a little bit, the content-wise, so that way you can catch up writing stuff without stopping the video. So hopefully you don't have to spend like 15 minutes taking notes. Keep that in mind, all right? So let's continue. So electrophoresis, what is it, right? So it's it's the separation method based on the applied electric field. So the substance that is electric, has electric charge would separate because it would move toward the the opposite electrode. So let's say you have a protein that is treated and would have negative charge that would be attracted to the positive direction. So again, it's based on electrostatic interaction. So, positive is attracted to the negative electrode and negative is attracted to the positive electrode. So keep that in mind. Now, so how does it work practically speaking? So now, in this case, you have protein that has, three-dimensional structure. So you have to apply heat to denature the protein so that in all this three-dimensional structure, protein folding, whether it's, it's a secondary structure, the hydrogen bonding within the protein like alpha helix, or hydrogen bonding to the, to multiple peptides like beta sheets, all this hydrogen bonding interaction, limits the availability of the peptide backbone toward the environment. So you apply heat, and then also you want to make sure that two substituents are not connected covalently. Like say you have, let's say disulfide bonds, like, you have two cysteine, and the sulfur and sulfur can form disulfide linkage. All right, so you want to make sure that there's no localized covalent bonds or strong interaction. In this case, ionic bonds, like, positive charged, and then, say something like, I don't know, let's say histidine and aspartate or something, like positive charge, negative charge, although, depending on the pH, it may not have positive and negative. But anyway, the point is that, if you have electrostatic interaction, under ionic, ionic interaction would come apart in aqueous environment anyways. But if it's a covalent interaction, like, or covalent linkage, like disulfide linkage, then you really have to chemically break the bond by applying reducing agent. So keep that in mind. Hydrogen bonding, you can come up, you can break it by applying heat, because boiling, if you boil water the 100 degrees or so, would be enough to overcome the energy barrier to separate the hydrogen bonding. But you would use beta-mercaptan ethanol. Mercaptan, by the way, is a thiol, just the old term called thiol. So it's SH instead of OH is attached. So the beta-mercaptan ethanol is one carbon has OH group. The other carbon, beta position, beta carbon has SH group. So keep that in mind. In terms of structure, let me see if I can actually draw this. So you have two carbons, and then you have one side, it has OH, so that part is fine. So you have hydrogen, hydrogen. This doesn't look great, but and then the other side has sulfur. All right, so something like this. All right, so beta-mercaptan ethanol, right? So that is used. So anyways, again, so this beta-mercaptan ethanol works as a reducing agent. It looks kind of not clear, but so be it. I'll go to the next slide so that this will disappear. Well, I guess not. All right, so let's see. But I'll come back to it. All right, so let's say you have a protein that has some kind of three-dimensional structure, and then applying heat would denature this protein, but then there might be some localized disulfide bond linkage, the covalently attached. So, your peptide may not go into the straight or, just into a denatured, fully denatured state because there might be localized covalent linkage. So you have to chemically treat it by, something like reducing agent, like beta-mercaptan ethanol. So in this case, beta-mercaptan ethanol, remember, you have SH groups. Let me draw this again. So you have C, and let me draw this slightly slower so that you would probably draw better. So you have OH group, all right? And then you have H, and then you have SH group, so SH, all right? So beta-mercaptan ethanol, so this hydrogen right here, right? You have two of these, all right? This one goes into this. So it's a reducing agent. So now another beta-mercaptan ethanol. Let me see if I can actually erase this. Hopefully I can erase this. Let's see. I find the eraser looking good. How about I erase this right here, right? And then another, let's see. Let me see if I can actually, I don't want to erase this. Let's see. I find a pen. So it can actually, another, the beta-mercaptan ethanol can form a disulfide linkage within, between these two beta-mercaptan ethanol. So then now you have disulfide linkage right here. So this bond right here would break, and then each hydrogen that used to be here, right, will be provided into here. So you have now the free thiol groups on this disulfide linkage is broken, and then you have cysteine substituent here and cysteine substituent here, all right? And then beta-mercaptan ethanols, two of these would make a again covalent linkage right here. So keep that in mind. So again, cysteine, I said cysteine, so let's just write it down. Cysteine, all right, has a thiol as a substituent, all right? So anyways, so now moving on, going back, so beta-mercaptan ethanol, right? Beta-mercaptan ethanol, right? Mercaptan again is a old term. It's called a thiol, right? So now we know that beta-mercaptan ethanol is used to make sure that all the covalent localized linkage is broken. So you have nice long the alkyl chain, right? So now I'm not alkyl chain, sorry, backbone, peptide backbone, right? So now, so SDS, sodium dodecyl sulfate. So why do we use this is the real question. Now, so remember this, this long alkyl tail part right here, right? This part right here, right? It's a detergent. So, so basically it has a, sodium, salt of, sulfate right here. It has a pretty strong negative charge. So keep that in mind. So, because of the long alkyl tail, all right, it has a, again, it's relatively, it's, they have electron density. It's it's a hydrophobic group. And then you have a backbone of peptide, all right? You have a backbone of peptide right here, all right? So, you have peptide backbone right here. So it's sort of like, the long alkyl tail lay along the, hydrophobic backbone of the peptide linkage. All right. Of course, you have amide, alpha carbon, and so on. Right. So you have these long alkyl tail lay next to it, And then, so at the end, the protein, in this case, peptide, accumulates, lots and lots of negative charge. So, technically speaking, 2 amino acids, per SDS. So if you think of like 10,000, Dalton, so 10,000 Dalton, or 20K Dalton, depending on how big your protein size of the protein is, whether it's a few thousand Dalton to 20K Dalton. Either way, proteins, it's 2 amino acids residues per SDS. So in either way, lots and lots of SDS, would lay next to these long peptide chains. So proteins accumulate lots and lots of charge. So it's a lot of negative charges. But then, negative charge would be attracted to the positive electrode, right? Because of electrostatic interaction. So all these negative charges would move toward the positive electrode. Of course, there are some positive species like, sodium, which doesn't really play a role in this particular case, but positive species, if they exist, then they go toward the negative electrode. So keep that in mind. Now, so separation based on the mass, of course, based on mass part charge ratio. So, but remember that proteins accumulate lots and lots of negative charge. So remember, it's like let's say 20K Dalton, right? So that means they have lots and lots of negatively charged sulfates. Whether it's 20,000 K or like 15,000 K, it's like a lot anyways. It's 5,000 Dalton, still a lot anyways. So we treat this denominator pretty large and pretty constant. So you can almost assume that proteins, the size of proteins, the species that are in the size of proteins, a few thousand to a few 20, 30K Dalton, would be like, you can basically, the separation would occur based on mass anyway. So keep that in mind. So proteins would migrate pretty much based on the mass, right? So keep that in mind. So now that said, right? So how do we do it? All right. So you have stacking gel, and that's where you load your sample. All right. So you have sample buffer. All right. And this sample buffer contains a couple of things. All right. SDS, glycine, and couple, and then some chlorides, salt of the buffer. Right. So anyway, so you have sample buffer, and you have stacking gel. You load your sample, and then you run the running buffer. All right. But also, running buffer and sample buffer both contains glycine, right? But in a different pH, especially in this stacking gel, the one you use is pH 6.8. It's an acidic condition, so keep that in mind. So how does it play a role, right? Glycine, remember, it's, you have, carboxylic acid and then amine group, but you have negative charge here and positive charge here. So that means, you have, this is, zwitter ion, right? So pH 6.8, right around pH 7 is where they occur as, these glycine occur as zwitter ion. But because it's slightly, less than 7, you would have, slightly higher population of the, positive species, all right? So, anyways, so, at lower pH, like pH 6.8, what happens? You have more positively charged, amine group. Of course, remember, pH 6.8, roughly, they do, this zwitter ion do a certain percentage of the, the glycine, does exist as a, zwitter ion. But also, there are, because it's less than the, zwitter ion pH, you have more of the positively charged glycine species as well. So keep that in mind. Now, because of that, all right, these glycines are protonated, which means it's positively charged, right? So the positive charge, all right, would, positive charged species would stay on top, right? So toward the negative electrode side, it kind of moves upward, all right? Move toward the negative electrode side, right? And then sort of like accumulates on top. Proteins, are negatively charged, so it tends to move downward, right? But so in this case, there's a separation between glycine and protein. So this, so glycine tends to accumulate on top, all right, as a cloud, all right, sort of like an electron cloud. So, it's like shielding the electric field. So you can see these arrows right here. These are, again, so electric field is not full strength because it gets blocked by these positively charged species. So now, and chloride, let's say, the chloride tends to move, down. It's small, and then it's, charge is large. So mass per charge ratio wise, it's pretty, it's a pretty good ratio, so that it tends to move fast. So chlorides tend to accumulate at the bottom, all right, near the positive electrode. So now you have positive species behind. So these are the same proteins, by the way. I just use the same proteins, all right? So imagine that these proteins are right in the middle, but then negative species at the, are at the bottom, and positive species are here. So now proteins don't experience as much electric field because it's shielded by these species, right? So keep that in mind. So therefore, the proteins don't feel as much electric field, so they migrate slowly. So although there are different sizes, they don't migrate, in a different, different speed, right? And also, gel matrix is large enough that that they are not like restricted. The mobility is not as restricted. So keep that in mind. So in this case, pictorially speaking, this is what happens. So you have glycine, behind the protein, chloride in front of the protein. So they all migrate right around the same speed. So keep that in mind. Now, so they migrate as an aggregate. So keep that in mind. Now, what happens afterwards? As soon as it reaches the resolving gel, so now you have resolving gel. So the gel actually continues like this, right? So protein enters into the resolving gel. So what happens, right? So now, so in resolving gel, pH is 8.8. So which means it's a basic environment. It's higher in the pH, all right? So which means glycine, exists as what? In this particular, environment, glycine is more basic in that basic environment, which means what? It's going to be deprotonated here, all right, and then deprotonated here. So now it would carry negative charge. It's hard to see the negative charge here, but it's negative charge right here, all right? So now the glycine is now carrying a negatively charged, charge. So it moves toward the positive electrode, right? So what happens now? You have chlorine that just goes down anyways at the bottom, but then glycine is now moved toward in this environment. Glycine just accumulates at the bottom. So in the back, you still have accumulation. So around if you have a gel here, I mean, if you have a gel, you do have accumulation of glycine on top of the stacking in the stacking gel, but now further you go out, less effective it will be, all right, in terms of like electrical shielding. So now proteins are now in this environment, but it's not experiencing as much it doesn't have the glycine right behind it, all right? So it would feel more electric field, and therefore it starts migrating in a different mobility or a different speed in resolving gel. So in this case, protein with different mass would migrate in different velocity, right? So because of that, and then also all these negatively charged species would just gush out anyways out of the buffer anyways, and then goes into the running buffer. So you don't have as much shielding, shielding. So proteins would experience much more strong electric field, right? And then they migrate in different mobility. So keep that in mind. So and so proteins would separate, right? So keep that in mind. Now that said, all right. So why do we care? Because, well, we can't see proteins, but we do have the molecular weight markers, which has colors. Oh, by the way, speaking of which, we do have something called difront. So in a sample buffer, you don't have to prepare a sample buffer. It's already prepared for you. but it has a dye front, which, it's basically based on bromophenol blue, which is a blue dye, which it's pretty small, it moves pretty fast. So that's the, it's the fastest thing that moves in the, in the gel matrix anyways. So because you can't see the proteins are always behind this dye front. So you can't see the protein, but you can see the dye front. So as long as dye front doesn't run off the gel, your proteins, it's somewhere, behind this dye front anyways, right? So keep that in mind. So anyways, this way, we can make sure that the proteins won't just run off the gel because we can't see the protein. We have to be able to see something, so that we use bromophenol blue, right? So keep that in mind, right? Now that said, all right, so why do we use dye front? Remember, each lane has a dye front, right? And because of dye front, we can actually calculate something called mobility. In terms of mobility, how do we do it? You just you measure the distance. So you just use the ruler and then just measure the distance from dye front, from where the gel starts to dye front, and then where the gel starts to the actual the molecular weight markers, right? And then you can calculate the RF value for each molecular weight marker protein, and that's a reference standard size protein. So you take a log of the molecular weight, all right, of the marker protein weight marker, and then you take a RF value in plot, and then you can just get the y is equal to mx plus b. All right, so you have different molecular weight standards, and then you plot based on log molecular weight versus RF value, and then you can get a straight line. So at this point, you have a relationship between high molecular weight and then versus low molecular weight, and how much RF value would correspond, right? And then you can get y is equal to mx plus b. Once you do that, and then you can measure the protein that you're interested in, right? The distance, RF value. Once you know the RF value for that particular protein, in this case GFP, you can just trace back, or of course, you want to use y is equal to mx plus b, but point is a relationship is established. So if you know this is your RF value for GFP, let's say, then you can actually calculate this log molecular weight. Of course, you want to do it more accurately by using y is equal to mx plus b, and then see, in this case, you have the you have the RF value. So you put the RF value in RF value, right? And then mx and x are both constant. I mean, in this case, you calculate based on y, like linear fit anyways. Then you can calculate y, which is log molecular weight. So keep that in mind, right? So now, so you have you can calculate mobility for each molecular weight markers, and then you have a protein. By the way, GFP, you don't know for sure if it's like, if it's one molecule, but it's two subunits. I mean, you can always look it up, but basically notice, I mean, so you may have to determine whether you have two bands or one band. So you want to make sure that depending on subunits that are connected with like disulfide linkage. So let's say you have one subunit, all right, and that goes into one peptide, and then another subunit that goes into one peptide. But if they are connected with disulfide linkage, but if you actually then they would migrate together. But remember, if you have two subunits are connected with disulfide linkage, now they would manifest as two bands. So you want to make sure that these two bands, you have molecular weight, certain molecular weight, but these maybe as a result of like one large molecular weight, which could have been here if they were in a disulfide linked protein, but it might have resolved into two different proteins. So keep that in mind. It could be, may not be. I mean, GFP could very well be just one subunit, one blob of protein. We don't, I mean, you can look it up, but anyways, so something to look, just to be careful when you actually run your gel. So keep that in mind, right? So, so again, So in this case, you may have, two markers, two subunits, but, it could be, two different weights, but it may add up to, just one big blob, right? Anyways, so keep that in mind. For those who are watching this video before the lab, after we actually finish the gel, running the gel, takes about 2 hours or so, and you're gonna cut, corners, as in like, you cut one side of the gel, or depending on which group, you can actually come, and cut these 2 corners, or, other group can cut 3 corners. And this way, basically, you can identify, even if the label is off, or, the lid, depending on where you label it. Gel, you cannot label the gel itself. So, you want to actually mark, where you cut so that you can identify which gel is, what. So, especially when you're like running 3 gels at the same time, you don't wanna like mix up the gels. So, you identify a, gel by, which gel it is by cutting multiple corners. So there are a couple of ways to do it. These are the 3 ways to identify, right? And also, 24 hours later or so, you come down and, destain the gel. So keep that in mind. So you're gonna have to destain the next day. So I just put group A, group B, and group C, or like 3, gels simultaneously running. You just, you just, do identifier cuts. So keep that in mind. All right, that's it. So in terms of like how to analyze gel, each lane by lane, I think I'm gonna do this a little bit in a separate video because it's already 30 minutes or so. So it's a pretty long video. I apologize, but I think, it might be, beneficial to do another video for analysis. All right, thanks for watching, and I'll see you in a day or 2 or so.